So this study focuses on nerve growth factor, or NGF. NGF is a patient-validated target for the treatment of chronic pain. There have been large clinical trials of monoclonal antibodies to NGF for the treatment of arthritis pain. And although NGF monoclonal antibodies were effective for treatment and effectively relieved arthritis pain in patients, they were not approved because of worsened joint damage in some patients. So we wish to explore more about the mechanisms by which NGF caused pain, with the intent of developing new treatments which could be devoid of these side effects associated with global sequestration of NGF with a monoclonal antibody. That was the rationale for the study. The study has identified new co-receptors for nerve growth factor, which are required for NGF to cause pain. And we have identified in this paper two proteins. The first is neuropilin-1. Neuropilin-1 was discovered in this paper as a co-receptor for nerve growth factor. It binds nerve growth factor with low affinity and brings nerve growth factor to the surface of the cell, and thereby facilitates its ability to activate a second receptor called TrkA. And TrkA is a signaling receptor which mediates pain. So neuropilin is a co-receptor for nerve growth factor. That's one thing it does. The second thing that we discovered that it does is it's a molecular chaperone. So it associates with TrkA and brings it to the surface of the cell, where it can be accessible to nerve growth factor. So it works in this twofold way. It binds nerve growth factor, it associates with a nerve growth factor receptor, and enhances its ability to signal. And so in the paper, we found that if you inhibit the function of neuropilin, and we've used many different ways to inhibit its function, we can strongly inhibit the ability of nerve growth factor to activate neurons, to cause them to become hyperexcitable, and to evoke pain in preclinical models. Now, importantly, we found that this process occurs not only in mice, but it also occurs in human nociceptors. So we have that translational component. We identified a second protein which sort of glues this complex together, and that's an intracellular protein called GYPSY1. So GYPSY1 is a protein that interacts with neuropilin, interacts with TrkA, brings them together, and allows nerve growth factor to more efficiently evoke pain. So we have two mediators, neuropilin and GYPSY1, and these are potentially new therapeutic targets for pain. Importantly, we found that these proteins are enriched in nociceptors. So they're enriched in the cells that detect and transmit painful signals. So by identifying targets which are enriched in nociceptors, we hope that we can avoid some of the side effects associated with systemic sequestration of nerve growth factor with a monoclonal antibody.